All right, we'll get started, right?
Yep.
So this is the first, I guess Friday was the first presentation you guys got for the BMRC summer program. Today, my purpose is to sort of talk about how we'd like to, a guideline for how we'd like the papers presented in Journal Club, which we'll be giving later in the summer. And this thing is broken up into two parts, kind of like what is a research paper in the parts, what their purpose is. And by paying attention to those details, well then I just have a short journal club that I prepared, sort of in the same style that I think it might serve as a good guideline for you guys. All right, so let's get started. Does this thing work? It does. There it is, Anatomy of a Primary Research Manuscript, an example of typical journal club. So on the right is a typical first page of a manuscript, peer-reviewed in a journal. This one's in Biomaterials, which is what I'm going to be reviewing today. I'm using the paper that I'm presenting today as my example here. And just some basic parts. That the header is, generally speaking, going to be the information about the journal itself and the publisher. The title, which is highlighted in green, is often a very descriptive, almost sentence-like title that describes is the message of the article according to the authors. Next comes this huge author list, which is getting longer in modern times because we are doing a lot more collaboration. How the authors are arranged in that list can differ from discipline to discipline. In our discipline, biology and so forth, generally speaking, the first author is the one who did a lot of the work in the paper. The last author is the person who kind of coordinated the research and often is the one who had the funding for that research. And these can be shared responsibilities. For example, Drs. Tang and Salazar-Puerta, they are equal contributors to this paper, and they're co-first authors, we might call them. And oftentimes there can be, Dr. Primasor is the PI of this paper, but there can often be other PIs also. And they're listed down the bottom box in red. The other key part we want to look at is the abstract, which is highlighted in brown. It's a brief overview of the entire paper. It includes the research question, the approach, the results, and an interpretation. Just to break this down, we want to look at the primary problem. Generally speaking, we want to define the gap in knowledge, say what was done, and the key findings. And you can see I've color highlighted these things. It's very condensed, and we're using some very specific but understandable words by people not necessarily in my field. They're scientists, but it's not specific to what I am doing. So then, this introduction, what is it? I think the introduction is the purpose is to say what's being studied and why. So you want to describe what you want to achieve, the rationale for why you're doing the study at all. There may be some theoretical framework you want to discuss to understand why you're doing the study the way you're doing and why it might be important. Present existing knowledge or recent developments in that field that puts this research in some context, okay? And then describe the significance and usefulness of this work. So obviously the introduction to this paper is about a page and a half long. I've summarized it in a very short paragraph, but it's not the whole thing, but the point is it kind of gives examples on being specific about, in this case, the problem of discogenic back pain and its frequency, and maybe there'll be some cost element, and then they'll go into some more mechanisms, for example. They'll talk about how you might do treatments for this thing, described in purple. You might give growth factors, you might give cells, you might overexpress some genes in this thing, and it gives the actual gap of knowledge. There's no direct translation to the human patients. There's really no treatments for this disease. And then they start describing what they think their treatment can be, which are the EVs as cell-derived carriers to deliver medications. And we'll go into this in greater detail. The materials and methods section describes how the study was done. It should be simple, precise, and very complete. The language used here is precise. That is, we're going to be describing exactly, for example, when you're doing cell culture, how long is the cells in culture, what media are you using, what temperature, what's the conditions this is happening. This is a relatively short description. She has other paragraphs, if you read the paper, where there's a whole paragraph describing the procedure. And the idea is I should be able to read that, materials and methods, and repeat your study as the researcher did and get reproducible results. That's its purpose. So new methods are described in great detail. established methods that may have been done for 10, 15 years now in the field, they may just give a very short description with any modifications they did, and then give references to where that original description is, so you can get the real detail on that paper. Then other things that should be in this materials and methods are like experimental groups. Who are the controls? What are their purpose? How are we using them? Sample sizes of each experiment, and the statistical methods you use to give the results and determine whether your experiment is successful and supports the hypothesis, yes or no. The results section is really the meat of your paper. It covers the main findings of this study. The data is presented in tables, figures, and associated legends. When we are in the results section, it's all about the results that you are doing or have produced in this study and nothing else. And you present it in as objective a fashion as possible. So you're not going to be using any adjectives to say how fantastic this work is or how amazing or how unusual, unless it really is unusual, which you might say, this is unexpected finding, and then give some very specific data about that data point. That's the only time I would just kind of give some sort of emotional response to my results section. So in general, this piece of the paper is the longest part, between 4 and 6 pages as a rule in most journals. I'll just describe briefly what we want to see and have maintained in a journal club. This is I actually like the figures in this paper quite a bit. This is an introductory figure sort of showing us in figure 2 what an EV is and its origin. When you're having a graph, you want to make sure that all the numbers and words and letters in it are legible to the viewer at the back of the room. So you want a pretty big font if you can get it. The axes are defined, and there is a unit of measure associated with that. And there's nothing more frustrating than saying y-axis is Fox expression. You have no idea what that means, what's it normalized to, or whatever. The significance in a paper is shown, in the asterisks here and defined down here. These are also really important, and oftentimes an n value will be given so you can understand how strong that significance may or may not be, even though they are defining it down here. Like, what does it mean? And often, so in a figure that and I'll show a figure later, we're not going to show this entire legend as is shown here. We'll pick out certain pieces like the n value, like significance, how it's defined. That would be probably all we put in there in addition to the graph itself. If it's a figure, again, I like figures to be as big as possible so you can see what's going on in it. But sometimes you really need to have some other information. It's very important that the figure, each panel, be defined. Like, what is it? And this does a nice job of non-injury, injury, delivery of control or PCMB6 EVs versus our experimental piece, which is the FOXF1. And we'll get into what that's all about later. And here you can see the scale bars are shown in every figure, which is not necessarily necessary unless it's different, which it is here. And she does a nice job defining what that scale bar means. And also, as you see here, she's defining the n value, so you can understand where the statistic comes. So there are graphs here that I'm not showing in this picture, where you can now appreciate how that number came to be and its strength. And then if there are things that she wants you to see in particular, there are arrows or arrowheads or lines, asterisks, and so forth. They should be defined clearly. Red arrows indicate the positive staining. And to the degree possible, you want to try to show all parts where that happens. And in the journal club, if that's important material, we want that in the journal club, and we want it to be legible and understandable from the back of the room. So then we come to discussion, which is where we finally get to discuss what our results mean. So we finally get to interpret them and specifically address how the results apply to the original question in the introductions. The introduction and the discussion are often parallel in many ways, and you're trying to refer back to your introduction. There should be no surprises. Your introduction should include all of the critical information that you'll be discussing in the significance of your work, if possible. So they are quite parallel. You want to show and discuss how the results of this original research apply to the hypothesis in particular and the field as a whole, putting it in context and even significance by comparing your work to previous work done. Generally speaking, you'll be looking at supportive work. I mean, I like to do both. And in a journal club, we want to hear both. But in an actual paper, people very rarely say, they'll say, my results differ, but that's because my results do this, and that implies that our results are better. But you have to be careful with that. That's where critical thinking really does matter. Because what we are going to be doing in a paper, what everyone does in a paper, is a collection of data, kind of like a house of cards. You build it like a lawyer. Here's my case. And you're presenting that case as best as you can. But there are clearly always holes in it. It's hard to have the perfect study. Most of us never really publish the perfect study. We have to acknowledge then in a discussion later, some other pieces might have, alternative interpretations might be discussed. The strengths and limitations of the study is a really important part. And oftentimes, when you just get to the limitations and things, you'll start discussing open-ended questions that your study results in, and there you can start discussing what the next step would be and what might be necessary to answer those questions. And she does a nice job of having all these sections in there pretty much. She does not really talk about alternative interpretations because journal editors and reviewers generally don't like you to cloud and muddy the water with alternative theories. So they want you to make a choice, stick with it, and let the reader decide whether they believe what you're saying or not, to some degree. So other things you're going to read at the end of a paper, which I think are important to think about. These days, so when I published my first paper in 1999, it was pretty much the pages you saw in the journal. That was it. Now, there's a lot more data to substantiate sort of how you got to your model, other things you did to confirm your model but aren't necessarily interesting scientifically speaking. Or if you're doing omics data, you have gigantic sets of data that can't be put in the primary paper, but you can provide links or small pieces in supplementary data. Critical is these days, if it's NIH funded, your data has to be shared. Oftentimes quickly online, or at least when someone asks you for it, you have to give it. You'll also see a section of acknowledgments for contributions that people and places have given to this paper that didn't rise to the level of co-authorship. And there's always going to be an open access statement that references this common attribution, which is how other people can use your data in their like review papers or talk in presentations and so forth, where they have to provide the original author and source. Other things that are more recent are the list of author contribution statements. So again, my first paper had 3 authors on it. My last papers have like 20 on it, and they want to know, okay, justify why 20 people are on this thing. And there are lots of roles. I have a little list here, roles. When you're doing your research, think about, who these people might be. Like if you were publishing something, who do you have helping you accomplish this work, and what are the different ways? So you have people teaching you methodology, you have the people doing the work with the investigation, you have the PI who conceptualizes the study in the first place and gets the money. Then there's data analysis, there's writing, editing. If you're a foreign language speaker and need to publish in English, oftentimes you'll get a service or someone to read your paper. They can be distributed here. And also project administration and data curation. Like this omics data are put somewhere up in the cloud, and someone's got to make sure you can search it and that it's available. And those people are often attributed here as well. There's in clinical, well, actually in all studies now, a conflict of interest statement needs to be made in some way, and an ethical review statement. So for humans, if you're involved in clinical studies, you'll be providing the IRB, which is the study protocol number, so that someone can go look it up. So this is also part of the curated data, right? So a researcher can look it up and find out whether the guidelines by which you got your data, are they acceptable? Which presumably, if they're IRB approved, they are. And did you do it right? If it's an animal study, you'll be citing the IACUC protocol, which is also reviewable, and probably some statements in the human case, adhering to the Helsinki Agreement, which is sort of the humane treatment of humans in studies. And in animals, you'll be adhering to the OLAW, which is like an IACUC principles, which are how to ethically treat animals in studies. And oftentimes, for 2 or 3 journals I published in lately, there's a whole 3 or 4 page questionnaire you have to fill out and demonstrate that you adhere to these principles. It's one sentence here, but it's actually quite a bit of work. So anyway, that kind of concludes my thoughts on what is a paper about. Go ahead. Questions?
Just to kind of zoom out a little bit on publishing and what this is all about. When you get money to do research, the ultimate goal is publishing your results and distributing that to other people. So if you do research and nobody knows about it, it's like a tree that falls in the forest and nobody hears it. So you want to disseminate your information out to other people. So journals used to be more like a magazine, an actual printed magazine with different journal articles in there. You can call them articles, you can call them papers. Now it's almost all electronic and accessible via PubMed and things like that. But an article or paper, what we're talking about, is the way that you distribute your results and kind of brag about what you've done.
Yeah, so the two outcomes people, your funding agencies, looking for are these publications or patents. Patents are also considered publications. And then secondary to that are presentations, things you might do. But that is your outcome at this level of research and basic research. So these things that we're doing, I welcome you guys to just interject a question or comment that you have. I realize I might not be clear, or I might skip over things. Please let me know when that happens. All right. So I just thought I would, as a segue, what is a journal club? What are you trying to do? And what we're trying to do is, all of us in this room, we're going to read this paper, two papers together, previous to this, and try to critically appraise this research and find out how we can use it for our own work in your own personal work or in the department as a whole. So functionally then, the contents of the paper are condensed. Obviously, we're not going to present all the words in this paper. Oftentimes, we define a length. I've had people define the length of your presentation as 10 slides in 5 minutes. We're going to go for 20 to 30 slides over 15 minutes. But up and down, some papers will require more and some will require less. So we attempt to present all the aspects I just showed on describing what a paper is in a very abbreviated form, as much as possible. And also, when presenting a paper, presumably we like the paper, and we're going to present this paper as though I did it. And I should know this data well enough to at least give you an educated comment on what happened in the paper and why I should believe it's true. I should also know why it might not be true, but I'm going to present it as though it's my data and that I know what I'm doing in it. So it's important to read those methods, understand those methods. And if you don't, and you find it to be kind of an ordeal to understand it, it is completely reasonable in the journal club presentation to have a slide about what this method is, and because it's likely that you're not the only person in this room who doesn't know what that method is. So like questions, most people will have the same question you have. That's true for reading as well. And it's always helpful to sort of like clarify something if it's a complicated subject. Okay, how am I doing time-wise? I have no watch. Okay, good. So then in the process of a journal club, this is a much more interactive process. So presumably, now I'm old school, I print my thing up and I got my notes on this thing. Maybe you guys have it on your PDF or whatever. You would be reading along kind of as this happens, and if you have questions, you just pipe them out, and we try to discuss it here and now. We give a title page, just like you would in the presentation. The important parts here is the title, which is conveying the point of this article, which in this case is the engineered extracellular vesicle-based gene therapy for the treatment of discogenic back pain, or intervertebral disc back pain. And we have our author list, and we try to specify, who our co-authors are, primary co-authors, and who our the principal investigators are, where it's located, a link or reference. In this case, it was a link. And that's what's really critical to this page, just like it is on the first page of the paper. In this case, we're going to be studying, so here again, my titles in these slides are descriptive. Like the idea of saying introduction as my title to me seems not really helpful. I want to know what the point of the slide is. Literally, I'm going to try and tell you that chronic low back pain is the leading cause of disability worldwide. I provide some data on that. In 2020, low back pain affected 619 million people globally, with a prevalence of 7.5% that increases dramatically over the age of 55. This intervertebral disc degeneration is a major cause of lower back pain, and it's often referred to as discogenic back pain. And epidemiological studies estimate that approximately 40% of cases are attributed to IVD degeneration. So my goal now is to define some of these terms, because my assumption is we're not all spine biologists. The most important structure we're going to talk about is the intervertebral disc. This is the joint between two vertebrae in your spine. The intervertebral disc is composed of three tissues. The nucleus pulposus, which I'll abbreviate as NP, the annulus fibrosus, and the vertebral end plate, abbreviated AF and EP respectively. Intervertebral health is maintained by homeostatic interactions between all these tissues, and we're going to specify here the nucleus pulposus and the end plate. And disruption of communication between tissues of the IVD plays an important role in the development of disease in this tissue. And I'm going to give you a figure here that kind of defines what these things are. Don't have a pointer. This is sort of diagrammatic look axially, and then from the side, we see our nucleus pulposus in the middle. It's very hydrated, very rich in arocan, and it plays the role of resisting compressive forces. Because it's so high in water, we know liquids are not compressible. It's only not compressible, or can help in compression if it's confined in a very fixed area that's not real flexible. And that's the role of the annulus fibrosus, a very specialized ligamentous structure that keeps the NP from expanding outside of its boundaries. And that's how it maintains its resistance to compressive forces. And then this piece here, very thin, is the cartilage end plate. I don't know how many of you guys have studied cartilage before. I knew cartilage as hip and knee, fingers, and things like that. It is not necessarily its structure is much more about distributing and handling forces, and you get nutrients in that through, like a sponge, you squeeze it. This end plate plays much more greater role. It's almost more porous in certain areas than others, and it acts as the only communication route for nutrients and metabolites to move in and out of the nucleus pulposus. So it's a really important structure. And what I'm providing here is just some images of what they actually look like. This is a healthy one, by the way. Okay, next slide. So then how do we characterize intervertebral disc degeneration? In general, we think that it's mechanical imbalances, inflammatory conditions, systemic inflammatory conditions like diabetes and so forth, might initiate this disease. It's characterized by loss of critical extracellular matrix components, which in this case are going to be aggrecan. It increases metabolic activity. These are enzymes that ordinarily are used in your body to remodel your tissues so that you can adapt to the forces that are present in your body. But here they go kind of haywire and break down the tissue too much, and it can never really recover. Also neurovascular invasion, so nerves and blood vessels start coming in via the annulus fibrosus. And this it contributes a lot to the pain that you might be feeling in this disease. So the big problem is, so here we're going to define what the big problem is. And we know that most therapies for back pain are really just palliative. I'm going to take things that inhibit pain, like NSAIDs and opioids. If things are acute-ish, you might get a steroid injection, which will reduce the inflammation. Sometimes these things are very beneficial, and you get one of them. Other times they start to contribute to disease. We don't know exactly why. But when pain dysfunction becomes so serious and none of this works, right now our only solution is surgery, which is not ideal. So we're looking for some sort of intermediate between it hurts and your ultimate surgery, which begins a cascade of surgeries you're going to have for the rest of your life, getting worse and more serious every time. So then she goes on to talk about the fact that biological therapies have shown promise in human cells, in vitro, and in some animal models. Three main types of therapy are listed here. There's the delivery of growth factors, which we know can promote the proper gene expression in these cells. Two of the prominent ones are TGF-beta and GDF6. There are cell-based therapies. So what do I mean by that? So in the late 90s it was described, well, we've known stem cells exist for a long time. In the 70s, we started characterizing what embryonic stem cells are. But then we discovered that adults have a type of stem cell also, which at that time was termed the mesenchymal stem cell or mesenchymal stromal cell. And people thought we can use that to build tissues. And then it became about regulating the inflammatory and regenerative process, which is all great. So the idea was, let's inject cells into the diseased intravertebral disc, and maybe they'll help. Then there's the viral gene therapy, where when a healthy intravertebral disc has certain, we'll get to this in a second, have certain factors that characterize that it's healthy, and then it has other factors that characterize that it is unhealthy. We can use viruses instead of the whole cell to deliver specific factors that kind of promote a much more global response. So for the intravertebral disc, the nucleus pulposus in particular, SOX9 has been shown, if you put that in the cells of the nucleus pulposus, you get a much increased expression of anabolic or beneficial genes to that tissue. But, she says, here's our gap in knowledge, direct translation is limited, because oftentimes when you deliver these things, you're causing disc injury. They have a limited time in which they work. Cell survival is pretty poor, especially with MSC delivery. If you're using iPS cells, you have the risk of tumors, and you have, in general, the problem of immunogenicity of anything foreign going into your body. You recognize it as other, and you killing it, and yourself in the process. Okay, so then I describe the previous work that applies immediately to this thing. Okay, so in a paper just before this one, they showed that a marker of juvenile intravertebral disc tissue was demonstrated to have the potential to drive cell reprogramming of diseased nucleus pulposus cells to a pro-anabolic or healthy state. Okay, so here I'm going to ask you guys right now, how familiar are you with the idea of a marker for a cell? Any thoughts on it? Okay, so I'll spend some time just defining what that is. This is a characteristic of a cell or tissue that defines that thing. For example, if I'll use a different gene, if I'm going to be a bone, my cell is going to become bone or is a bone, RUNX2 is a master regulator of what that thing is and characterizes what that thing is. So we will call that a marker. In the case of RUNX2, we know what it's doing. But there are many markers for which we just know it characterizes that cell and defines that cell. We might not have a function. We might not even know what it is. And we call it something like factor X, which is where a lot of these weird letters and numbers come from, because when we first discovered it, we had no idea what it was. Then does the phenomenon of cellular reprogramming, does that mean anything to you guys? So a stem cell, I should have had probably a slide about this. We're growing. Stem cells are cells defined by the ability to self-renew while also providing a group of cells that can become differentiated tissues. And you have different levels. You can have a unipotent cell, for example, that exists in your skin that gives rise to the keratinized cells that are your skin. You can have stem cells that give rise to all the cells of a certain germ layer. Does the word germ layer mean anything to you guys? Okay, so germ layers. It's okay. The ectoderm, which gives rise to my eyes, my skin, fingernails, all that is ectodermal. That's one of the three primary layers in an embryo. All this stuff from my mouth to anus on the inside, endoderm, is another layer. And, stomach, liver, or not liver, the canals of liver, your intestine, so forth. These are endodermal. And then the bones, muscles, blood vessels, and so forth, that's mesoderm. Generally speaking, differentiation is, for cells that I would get from me, are restricted to one of those three things. Can I be a skin? Can I be muscle and bone? Can I be gut lining in some fashion? But an embryonic stem cell can become all of those things. We call that a totipotent or cell. Normally we would get those from an embryo, like an 8-cell embryo or 16-cell embryo, very early on. A gentleman in Japan, Yamanaka, pulled together research over many years and transfected an adult skin cell with 4 factors called the Yamanaka factors after him. They are OKSM. We can go into this, what this means later. It's OCT4, KLF4, SOX2, and MYC, c-MYC. These 4 factors, when put into an adult somatic cell, so differentiated, return to an embryonic totipotent state, can contribute to all germ layers. Why do we know that? If we take that cell and inject it into a mouse, that cell will locate somewhere in the body and produce a tumor. And that tumor has representative tissues from all germ layers. So it has gut lining in it. It's got muscle and bone in it. It's got eyes and hair, sometimes teeth. I mean, not organized in a mouth or a body, but they're like all over the place. So that's how we define it. And I think earlier I mentioned the word that some of these cells could be tumorigenic. That is the example of what can happen there. Anyway, so does that sentence now make more sense? Markers of healthy juvenile IBD tissue demonstrate the potential to drive cell reprogramming of diseased human NPs into healthier ones. Does that sentence make sense? And you can always ask questions that will help me make a better representation for you. All right? So this is then more specific. I'm going to get into what an EV is in a second. But these extracellular vesicles, so these are packages of material that a cell makes and sends off into the environment. If the cell is expressing a marker of NP, which in this case is two genes, either brachyury or forkhead box, FOXF1, which are markers of embryonic healthy NP, they can induce anabolic properties. These EVs that are derived from this cell, produced here, are isolated and come to another disease cell and can help these cells become more healthy. So this is their idea then, that EVs can be used as a therapy or to deliver therapies. And this is some primary data they showed in a previous paper where these EVs, when delivered to a disease cell type, can increase the markers in that disease cell type of anabolic markers, and also some functional things like KRA19 and SOX9, which are known to drive anabolism in nucleus cells. So that's the previous data I want to talk about. Then what is this EV thing we're talking about? I just mentioned these are vesicles that are derived from parts of the membrane of a cell that carry a whole slew of factors from itself to its neighbors. So I guess when I was doing cell biology in 1988, I conceived of things like TGF-beta and enzymes being sent off alone, kind of in solution, and moving through the world kind of randomly, or I don't know exactly how it worked. But here we have our idea is the cells have a whole program to deliver to somebody, and they have a whole collection of molecules that contribute to that in this vesicle. So they are defined by size, the 50 to 200 microns. They have a wide variety of things inside of them, proteins, mRNA, including microRNA, nucleic acids, and other things, including the composition of the lipid itself. The receptor cell has specific receptors. The receiving cell has specific receptors for these EVs. And an EV can have a specific function depending on the state of the cell from which it's derived. So these are important things to think about when we're using EVs. And she does discuss this in her paper at length. The goal of this project, when we want to see this, is to engineer EVs that can deliver developmentally specific transcription factors to degenerate, painful IBDs. And they hope that the EVs can mitigate functional and structural changes that are associated with this disease state. And we'll get into what that means in a second. They do not explicitly state hypothesis. I think at our level, and even sometimes at my level, I like to express the hypothesis, which is always a positive, it's an active voice thing. So it's not could this, could that, may that. It is this or is not this. So what I want to say here, I think the overall hypothesis might read something like EVs carrying the developmental transcription factor FOXF1, which is a factor that drives maturation of nucleus pulposa cells. These EVs will reduce IBD structural, biochemical, and biomechanical changes in murine, lower degenerative model, and reduce the pain behaviors in these affected mice. So she doesn't actually say this. I think that her introduction is clear enough that you would intuit that this is what she wants to do. But in a journal club presentation, if you can, struggle with what that hypothesis statement is, because it does make life easier for all of you, if I can distill it into one sentence. Often in the introduction, we also want to know the experimental design. What happened here? She does a great job of having a pictorial description of what that experimental design is. So I've just pasted that diagram here and follow what it says on the left, just outlining for you, the reader, on what she's doing. So first she's going to make the EVs and show us what the EVs are. She has two kinds of EVs. She has one that's going to be made by cells that are overexpressing FOXF1, this nucleus pulposus factor in healthy and juvenile NP. She's also going to have her control, which is defined as the PCMB6, which is the same plasmid in which the FOXF1 is overexpressed, but just without that part. She cultures these cells for 24 hours, and then she isolates the EVs. And I think I don't have it listed here how she did that, but she does it with these columns from Thermo Fisher. So she's collecting these things based on size exclusion, so how big are these things, and then centrifugation, which are typical combination of things you do to isolate EVs. Then she's going to test the FOXF1 EV efficacy in mitigating IDD pathogenesis in an actual in vivo model, where she's injecting 3 consecutive discs, as you see here. And then her outcomes are briefly listed, and we'd like to see them. She's going to look at behavioral analysis, which I'm going to get into. She's going to use some medical imaging, which includes CTs and MRIs. I think just MRI. We'll see in a second. Some functional outcomes, biomechanical. So the spine has a biomechanical function in my body, keeping us upright and moving. We want to find out if that's changed. And then some histological and immunochemical data of the tissue itself and the cells themselves, characterizing what they are doing. And then we always want, if possible, the paper should be very clear about what our experimental groups is, so we can understand what are the scope of this data. So she describes these experimental groups, the sham surgery, where I guess the skin is open, we don't actually do a puncture. We do a puncture, which is going to cause the disease. We do a puncture and deliver EVs that we expect to have no effect, which are from cells that were transduced with PCMB6. And then we have EVs that are from the FOXF1 overexpressing cells. Those are our 4 groups. It was injected in 3 consecutive discs, so we have lots of samples to choose from. They can be used in different purposes. For example, you might do mechanical, biochemical, and imaging on all 3, for example, and get a pretty good end value. She has her time points when she did the studies, up to 12 weeks. Oh, and here she defines what each segment was used for. There it is, motion segments, 1 second gag content, and then histology and chemistry.
I'm just curious.
Fire away.
I don't really know that much about spine anatomy. Wouldn't each vertebrae have different biological properties?
So you are right. There are things in common between all vertebral bodies, at least within the cervical spine, thoracic spine, lumbar spine, for example. There is a strong argument made that each, by segment, even within an anatomical region, there is variation as you move. It's clear. The structure of vertebral bodies change, the size of these connective tissues change, and how much of certain things is changing. So she's going to be comparing L3, L4 for this one type of analysis across all her animals. L5 to L6 is analyzed the same way across all animals. She's not going to be necessarily comparing what's going on 3, 4 versus 5, 6. That's a good question. Any other questions about it, the experimental groups or anything? So then she says, and we want to hear this as well, think carefully about what the statistics are. Now, I am not a great statistician, but I do want to point out that she is using an appropriate rank test for the behavioral analysis and an ANOVA to look at multivariability. And then for the structural and functional assessments, in general, it's normality, Wilks test with normality, and a one-way ANOVA, which is typical. And she defines her alpha, right, and her p-values. So then we will go into our results. You could put all the results in your journal club. I don't find that useful. First of all, the images are really small and will take forever to go through. So I try to distill what I think are the important points of this figure, provide what I think is most important here, and give it to you in one succinct slide, if possible. You, with the whole paper in front of you, if you have questions on a particular panel, you ask it at this point. Anyway, I would say that this figure is really about characterizing the EVs that are produced by these FOXF1 transfected cells. They demonstrate in part F that the EVs made by the controls, PCMB6 versus the experimental FOXF1, are not exactly the same, but quite similar in size. She then shows that FOXF1 expression within the cells that are making these EVs is in fact much increased compared to control. So we know that the source cells are doing what they're supposed to do. And the EVs that are being produced by these cells actually are carrying different things. I mean, this is a very, I don't think this is the Western as shown, but there is a little bit of variation. The point is they're not carrying everything that the mouse embryonic fibroblast is carrying. I think it's important for just a second to just, these embryonic fibroblasts, what are they? I've only produced them from chicken. I used to work in chick biology quite a bit. What these are akin to our adult stem cells in that you basically take the body, eviscerated body of trimester 1 embryos, chop them up into small pieces, digest them for a limited amount of time, 5 or 10 minutes, strain them, and whatever cell comes out and lands on plastic, sticks there, and divides, that is your embryonic fibroblast. So it is not defined by genes expressing or anything like that. It's, is it quickly accessible by enzymes from tissue? Does it adhere? And does it clonally divide? That's what ends up on the plastic. And that's exactly how adult mesenchymal stem cells were isolated as well. To the point, it was so poorly characterized that today we would no longer call them stem cells anymore because it was, that characterization is so poor. But we still use the terminology. Anyway, so in the next figure, so now we're going to get into the results. And we try to just, again, we're trying to describe these figures in clear sentences in the title. So FOXF1 and PCMB6 EVs can modulate pain behavior in a mouse LDP model. And I'm showing here an example. So this is the grip time. They did 4 kind of experiments. I probably should have outlined them for behavior. One experiment, they dropped the mouse into an open box that's dark and quiet. And they look at the animal for a certain amount of time. I do it for 10 minutes. I think she does it for 6, but I'm totally sure. What you're looking for is, what does a mouse do? A mouse that is happy explores, it sniffs, it preens, it does all kinds of things a normal mouse does all the time. If it's in pain, it may do that less or more. And this is something that's still being worked on. But we experience pain, and it changes our behavior as human beings. We believe that should happen for mice too, and she's asking whether that's true. In this case, she can see that grip time, which is, in this case, she's doing a grip test. So here, animals are, you have like a grid, a wire grid. You put the animal on the grid, shake it a little bit, turn it upside down, and ask, will it drop in the next 30 to 60 seconds? And you count, you can record when the mouse lets go. That's the grip test. In females, you can see there's an increase, a decrease in the time that can hold on at 10 weeks. And in males, it happens at 12 weeks, it happens with significance. So she here is in the graph, she's defining significance, what it is. This is not quite reaching significance at 0.054. This is, or not quite, 0.065. So she's saying that she believes this is trending. She wants to say it, but she wants to tell you that it's not significant yet. I think that's valuable. She does a good job doing that. And then it seems like the FOXF1 cells are improving things. Some behaviors, like here, cold hypersensitivity. So what is this test? We've got a cold plate. We're going to drop the mouse down on it. And we're going to film it for a minute. At some point, it's going to be so cold, it's going to pick up its feet to get off the cold surface. There's an idea that if you're in pain in general, you're going to be more responsive to a painful stimulus than if you're not. So your threshold is lower when you're already in pain. So they should pick up their paw faster. And here she's telling us that there isn't a whole lot of change in females, but there is in males. So at this behavior, it's sexually dimorphic, which is interesting because we're still trying to learn as a field, how does animal behavior, how is it reflected in pain? And here we're saying that the male and female, for the distance traveled, so we film at the same time. So in the open field test, we drop the mouse in there, We just film what goes on. And we count how far does it go? How fast it's going? Does it ever preen itself? Does it ever explore with its nose? Does it ever rear up on the walls? These are all behaviors that are significant. Both males and females are affected to some degree by the FOXF1 EVs in a positive manner when it comes to the total motion, the total distance traveled. So summarizing, we didn't look at all the figures, and if you had questions about any particular panel, you would ask me now, and we would review it. I got the panel here, we can find the paper. Behavioral testing suggests that FOXF1 and PCME EVs have the potential to modulate pain behaviors in mouse lumbar disc puncture model 12 weeks after treatment. And that, interestingly, sex can influence which outcome is changed. As example by the cold plate response, for example. So then she gives us, one of the key factors and what he measures in intervertebral disc health, and whether you have disease or not, is in an X-ray, is it narrow or not? And this is true for your knees too. When you go to check, when your parents go to get checked for knee OA, they're probably gonna take an X-ray with the person staying there, and all they're asking is your space more narrow? If it's more narrow, the bones are closer together, you probably have cartilage degeneration, your joint space is less, and you probably have osteoarthritis. Same goes for spine. I am just showing you here, this is the T2, I believe, which in this case, water is white. And we know our intervertebral disc has a lot of water, right? 'Cause it's resisting compression. And the discs are shown here as the arrows, white lines. And she's asking how white is it? And she sees an improvement with FOXF1 versus her control. And that it's approaching, well, not completely the same as the non-injury state. This graph comes from another set of images not shown on the slide, but I'm just trying to say that its disc height, which was shown in T1 imaging, is also seems to be improved by the FOXF1 EVs. It is a joint after all. I'm not going to go into a whole lot of detail here, but what the test essentially is you isolate this one segment, and you're going to load it. There are many loading regimens for determining mechanical properties of a tissue. In this case, they're using what's called dynamic. So they're going to press this thing over and over again. Critical to this is they're going to make sure the machine is delivering a load and also sensing the response or sensing the forces applying. In this case, yes, we could have amplitude be exactly the same, but we're not. We're going to make sure that the force is always the same. And what happens is every time you press it, there's a little bit of deformation of tissue, and you're going to be decreasing the amount of force that's needed to get the deformation you're looking for. In the end, you get a graph like this. And all she wants to show you is she can determine things like stiffness and range of motion by looking at the linear area. And at both ends, there's another linear area that can help you with determine what the compressive stiffness is versus the tensile stiffness from one experiment done over, I think it was 16 minutes. And this is an example then over just looking at the stiffness, which was a measure I often use, that the FOXF1 EVs seem to really improve the disc versus the untreated and the controls. Seems to. Then we're going to look at the tissue itself and find out whether we can see differences. So I've paired two things here. This is it's one row of a whole page figure showing us Alcian blue staining. Alcian blue staining stains proteoglycans that are specific to articular cartilage and the intervertebral disc. Especially at pH 1, which is where this was carried out. So we can get a visualization of the gags that are in this disc. And she also, other sample, as you remember the second sample, is used for something called the DMMB assay, which is a quantitation of those gags in a biochemical manner. So you read that with a spec once you process it. I think visually, we can see a lot less blue versus control. And with treatment, that looks pretty much, we're approaching normal. DMMB, so the degeneration score using this thing, so there's a whole scoring mechanism, which they simply say, we reference this thing. You gotta go find that paper and see what that scoring system is. This lab has tried to innovate a new scoring system that's more complete in terms of spine. It's not used here, even though she's a collaborator on that paper, whatever. And then this data is supported by the GAG assay. And I thought the pictures are more exciting than the graph of the GAG, so this is why I put that here. And then she's also going to show us that FOXF1 is actually induced in vivo. So she does immunochemistry for FOXF1 and in the five groups, right? And she points out what the signal is. And I think she has a graph that I'm not showing here to show that there are more FOXF1 expressing cells with the FOXF1 EVs as opposed to the CMV6, which I think you can see here, for example. So in summary, it seems like EVs engineered to carry this genetic cargo for a developmental transcription factor such as FOXF1 to mice IBD joints in vivo. They have the potential to repair the structure and function of the degenerative tissue while mitigating the pain behaviors, maybe. Engineered EVs carrying FOXF1, they reduce some pain-related behaviors, demonstrating some sex dimorphic changes. They preserve disc hydration and height, which you saw by the CTs. They maintain motor unit disc biomechanical properties, which you saw by the biomechanical testing, and they preserve disc histomorphometry and GAG content. That's their summary. They do tout their work. Why does this matter? This is the first study to deliver developmental transcription factors to a diseased joint using EVs in vivo. And they combine the biomechanical testing of IBD joints with imaging, ECM changes, and pain behaviors to confirm that this matters in a functional way for the animal. And it could then serve as a therapeutic intervention. They do a little discussion on the limitations of the study. And we want to see what those are. Though the disc health was improved with EV treatment, FOXF1 EVs rarely performed better than control EVs. So the significance was not always there. The trend was, but not significance. FOXF1 is a transcription factor of the nucleus pulposus. The immunochemistry in figure 7 focuses on FOXF1 localization in cells of the annulus, and I don't understand why. The therapeutic treatment is administered at the time of the injury, possibly speaking to a preventative agent in IDD. However, the study does not speak to the ability to reverse advanced degeneration, which is how you and I will encounter this therapy. We're not going to go there at the moment of injury. We're going to go there once we have a problem. And the mice that were injured were 15 weeks of age, which is a young adult. Again, most of us are not going to have this problem as young adults. We have this at 50s and 60s and beyond. So regeneration may be reduced or insignificant in aged animals. And that will be all the components, trying to summarize the parts of the paper in a short presentation. I think that my journal club was about 20 slides long. It's planned to be about 15 minutes long. If you guys ask questions, it can easily go 40 minutes or 30 minutes. And that will allow us to have 2 journal clubs a week if possible.